I'm Sherry Lundeen, National President of ARCS Foundation. On behalf of the ARCS Board of Directors, I would like to welcome all attendees to the first ever ARCS Scholar Panel. Since the COVID-19 global pandemic brought a stop to life as we know it, we are proud to see many scholars applying their talents and knowledge in new ways to help address the crisis. Necessity is the mother of invention, and never before has this proverb from the Greek philosopher Plato been more pertinent. Today, you will be inspired by 4 ARCS scholars and alumni who are responding to the current national challenge in remarkable ways. While they are making valiant efforts in the fight against COVID-19, they are not alone. In the coming months, you will see other scholars and alumni highlighted on social media and in Archives, our e-newsletter. All are making outstanding contributions working to aid the United States and its citizens in the battle against the virus. This is a live moderated panel. We will have time for questions following the presentations. You may submit your questions at any time by clicking the Q&A function at the bottom of your screen. We encourage you to tweet and post about the event today. Be sure to include @ARCS Foundation when you do so. I would now like to introduce our moderator, Mr. Bill Liss. Bill is an award-winning reporter and journalist who is no stranger to ARCS Foundation. He is married to ARCS Atlanta chapter member and former chapter president, Debbie Liss, and together they have supported numerous ARCS scholars for 2 decades. He has been named Georgia Broadcaster of the Year, earned almost a dozen Emmy Awards, received the Edward R. Morrow Team Investigative Reporting Award, plus many other awards. Bill loves to travel and is a member of the board and head of media for the Worldwide Explorers Club. He has been on expeditions to the North Pole, Mount Everest, and the Arctic, to name a few. As a friend of Bill's, I can tell you many more things about him, but I will stop by saying, as a reporter and moderator, Bill does his research, and I know he has been preparing for today's panel for many weeks. Bill, it is a pleasure to have you with us today.
Well, Sherry, thank you so very much. And Nancy Chambers, of course, who put this all together. My great thanks to you for all you've done. And I want to say good afternoon to our most distinguished groups of ARCS scholars, both on today's panel and joining us from around the country, and also to our most distinguished attendees, the national membership of ARCS. Now, together you will represent the very best of scientific innovation, scientific discovery, and whether you act in a capacity of creating those innovations and discovery, or as one of their benefactors, your roles all meld into one, advancing science and enhancing our magnificent younger generation, 4 of whom you're going to meet today, to set standards and goals that many of us, frankly, never dreamed possible. Then, as we all join to salute scientific achievements of our ARCS scholars, we suddenly face a worldwide bombshell, COVID-19, spreading its deadly web around the globe. And for many of our ARCS scholars, the COVID-19 epidemic presented extraordinary challenges and unique opportunities to harness scientific skills and exceptional intellect on a fast-moving yet most inhospitable track. It turned those exceptional skills from long-term research and development to inventing COVID-19 diagnostic and life-saving equipment, to enlisting an army of COVID-19 diagnostic and life-saving people who are now out protecting thousands of people in Hawaii, and to a simple dry swab test that can save millions of lives. These ARCS scholars are with us this afternoon, and if you look up at the top of your screen, you will see them all. And I'm going to start with you, Alexandra, or may I also call you Sasha, which I think I can. You and your colleagues are the inventors of the mysterious Ventivator. That's quite a name clearly associated with Darth Vader. Can you tell our audience around the United States, what is it that you have come up with and how?
Absolutely, Bill. So initially when the COVID-19 pandemic hit and it became clear that we had an unmet need, we could potentially be running out of ventilators, me and some of my colleagues, a great team at the Biomems lab at UC Irvine, built this ventilator, and I'm going to show you. It's a portable, low-cost ventilator. In fact, I'm holding it here. And it's a pneumatic ventilator that runs off of the compressed air lines that are available at your hospital bedside or even a portable compressor. And basically it works because we have these two very simple solenoid valves. I don't have it hooked up here, but you have an inlet valve, which is going to compress an ambu bag, and you have an outlet valve that allows the air to exit the chamber and release your ambu bag. And by controlling these valves, we can actually control exactly how much volume of air to deliver to a patient and the time of inhalation and exhalation. We also have put in a number of sensors, pressure sensors, flow sensors, to be able to also control the pressure that a patient is delivering. And our initial concept ran off of a very simple Raspberry Pi, so something off the shelf. But right now we have this amazing team of programmers that are developing an Android and iOS app for the ventilator. And we also have a member of our team developing a mechanical interface, so in case you don't have some access to digital technology, it can just be run manually as well. And going forward, the focus is actually to ruggedize. It looks a little rugged already, got a big handle here, but we want to ruggedize the ventilator, because what we want to do for the ventivator is we actually want to use it in what we call extreme point of care environments. So basically, these are rural areas in developing countries which have little to no primary care infrastructure. And these areas in places like India, places like Africa, we have a lot of contacts there. In these places, chronic respiratory diseases are one of the leading causes of death, even before the COVID-19 pandemic. So going forward, we are going to have a group of students at UCI partnering with several contacts we have in India in particular to basically ruggedize the ventivator and make it something that can be used in these extreme POC point of care settings.
Well, a couple of things I want to ask you, Sasha. First of all, how did you come up with that name?
Well, so you can go on our website, ventivator.com, and you can watch some of our videos. And the sound that it makes when it's breathing, I like to say, sounds a lot like Darth Vader. He did have a mask with probably a built-in ventilator. So it was kind of just a fun little name that we came up with.
Well, one of the greatest controversies in this country has been receiving ventilators and having enough ventilators to go around. This sudden surge of COVID-19 forced hospitals to take whatever they have and sometimes double up. And I notice in some hospitals, even as you know, they use ventilators that were pumped by hand in order to get them to work.
Yes.
How available is your ventilator going to be, and how would you compare it to the multi-$1,000 cost of the regular ventilators? I mean, have you come up with something, I assume, quite revolutionary?
Well, I think that there's a number of people right now, as you said, when this pandemic has really generated the ability for a lot of groups to come up with these types of low-cost ventilators. So there's a number of other groups also working on similar designs, different mechanical methods of actuating this ambu bag. And ours is a pneumatic one. Ours is actually, right now, all of our components are less than $150, and that's off the shelf, pricing. So if we are actually looking to partner with some larger companies for manufacturing, bring down the cost even more, because when you look at these extreme point of care areas, they don't have access to a lot of the same infrastructure, the same laboratory and hospital infrastructure. So we really need to get those costs down to a fraction of what they are right now. And hopefully, even if it's not available for the next weeks or months, hopefully in the next crisis, we can be prepared, because we have this surge now of these low-cost devices that people have been working on.
Well, how effective would you say, I want to get into the other thing you're doing too, but how effective is your ventilator compared to the ones that we're hearing all about and the ones that are in the hospitals now?
So we haven't actually had the chance to test this on patients, but right now, you know, we can, the thing that is unique about our ventilator compared with a lot of the other low-cost ventilators is most of them can only control the volume, the tidal volume that is delivered to a patient because they are mechanically actuated. But our ventilator, we are developing the ability to be able to control not only the tidal volume, but also the pressure. So the fact that we have several of these sensors in there is quite unique, because a lot of these $30,000 ventilators that are used in the hospital setting, they need to be able to precisely control both tidal volume and pressure, because depending on exactly the respiratory condition that a patient has, they may be okay with just having volume control, but sometimes you really need to make sure that you have the proper pressure control not to blow out the lungs.
Well,
So
can we also
that's
ask you now, I'm sorry to interrupt you, but we want to cover too, your
Yes.
diagnostic testing. Can you tell us a little bit about what you have been part of and the team that's put this together? And you
Yes.
mentioned 67 viruses. That's extraordinary.
67 antigens or antibodies. So I will tell everybody about it. I'm working at a startup point of care biomedical device company, Sensor Kinesis Corporation, which was started by, founded by an inventor, Josh Shohar, and also Dr. Roger Kornberg, who's a Nobel laureate. And for a while, we've been working on this portable biosensor platform. And when the pandemic hit, we kind of shifted to use our platform to detect COVID-19. And right now we have kind of two directions, two submissions. We're actually starting our submission to the FDA over this next week. So one is for just direct viral detection on our platform. And the other one, what you had mentioned, we're partnering with my PhD advisor, Mark Madu at UC Irvine, and also Dr. Phil Fellner, to develop a diagnostic that can detect 67 different antibodies, immune system antibodies for common respiratory viruses. So not just one antibody for COVID-19, but we have 35 antibodies just for SARS-CoV-2 and other coronaviruses, and then up to 30 other antibodies for things like influenza, adenovirus, etc. And why this is so important is not only does it tell you whether you've been exposed to COVID-19, but it can give you this personalized, I would say almost an immune system fingerprint about how that infection affected your immune system. So on an individual level, this is important because it can inform treatment and make sure that someone is given a personalized treatment that is right for their immune response. But then on the community level and the society level, it can begin to solve the puzzle of why certain groups are affected differently by this virus than others. And we're actually partnering with Oracle to do a lot of the data analysis on that response as well. So it's very exciting.
Very exciting. Very exciting. I got to tell you, that's wonderful. And now I'm just going to switch gears if I can. And Serge, I'd like to ask you, you know, it's often said that it takes a village to make something happen. Well, for you and your entrepreneurial skills, it didn't take a village. It took an army, a volunteer army. Can you tell us about what you've accomplished, what you're doing, and how it's impacting the population of Hawaii? Well, I just found out now that the governor has extended it, that if you're coming to Hawaii, so anybody out there in the audience who's got tickets to head to Hawaii, be a little bit careful because you're going to spend 14 days in quarantine before you can get to the beach and really enjoy yourself. Serge, can you tell us what you've been doing? It sounds quite exciting.
Absolutely, Bill. Thank you. Aloha from Honolulu. My name is Sergey, and I am the founder of the Aloha Mask. We're a community organization dedicated to providing cotton face masks to frontline workers, and to date, we've produced 10,000 face masks to help our community. The island of Oahu is just 44 miles long, and it's located 2,000 miles away from any mainland US. Our healthcare system, while excellent in its own right, is equally small and was by no means prepared to this pandemic. I made my first face mask on March 20th, 10 days before the Honolulu mayor issued a stay-at-home order. My first thought was just how long it took to cut the fabric for this face mask, and I experimented with a few manual fabric cutting methods, but they all had their drawbacks. That is until I tried cutting cotton using the industrial laser cutter. Suddenly, I was able to cut patterns for 100 face masks in a single day. March 25th, I delivered the first batch of the face mask that I and my neighbor put together to a local grocery store. With the industrial cutter at hand, I quickly realized that there's no way for me and my small band of sewing volunteers to assemble all the face masks we could produce. We needed to organize, we needed to establish a network of volunteers across the island and secure the stock of fabric. And so Aloha Mask was born. I quickly bought the alohamask.org domain and got in touch with Patrick and Brooke Jones, veterans of many volunteer campaigns. As we scaled up our production, our team grew, and none of this would be possible without Aloha Mask internal team. While I and another volunteer student, Christopher Ketter, handled the manufacturing, the rest of the team handled everything, from developing the safety guidelines to our volunteers, acting as hospital liaisons, organizing weekly routes for our drivers, as well as working with local businesses and volunteer organizations to secure stocks of fabric. Our team was also able to secure the blessing from the mayor that allowed our drivers to travel the city unimpeded during the lockdown. With the internal team assembled, the news of our effort reached the local broadcasts. Overnight, we had 450 volunteer sewers and 50 drivers signed up to help our effort. Local businesses and institutions began donating fabric to our cause. As time went on, we got more efficient. The first week, we produced just over 300 face masks. The second week, we produced 1,000. The third, we pushed that number to 1,600. News of our capabilities reached other sewing circles. Soon, besides cutting fabric for our volunteers, we were processing fabric for the University of Hawaii health partners, Kapiolani Women's and Children's Hospital, UH Theater, as well as local volunteer groups. If you need a team of dedicated sewers, visit your local theater. After many years of experience creating sets and costumes, our university theater department was able to produce many thousands of masks for our cause. The most rewarding part of this experience was watching the island of Oahu respond to the COVID-19 crisis. I rarely interacted with the sewing volunteers, but I met every one of the delivery drivers. Regardless of sex, race, or income, I saw the entire community come together to support the people on the front line of the pandemic. From bike messengers doing local routes to professionals driving to the furthest reaches of Oahu, every volunteer was eager to help of our cause. We never had a face mask go out to our sewers that didn't come back to us. The COVID-19 pandemic and the lockdown took a heavy toll on all of us. And I would like to think that the Aloha Mask help those who participated in their feelings of loneliness and hopelessness while they're hunkering down in their homes. While we were watching the events unfold in New York, we were preparing for the worst. With the N95 mask delivery to our island uncertain, we took guidance from our colleagues at the Marine Science Department at the University of Hawaii. They were using state-of-the-art atmospheric equipment in testing various materials as ad hoc filters. The materials that we settled on was the H600 surgical wrap, which compared favorably to the N95 masks commercially available. All of the masks that we produced had a filter pocket which could easily incorporate a makeshift filter, and we produced 1,000 filter elements just in case the worst were to occur and our masks were to end up in the intensive care units. In the end, Oahu got lucky. Quick action of the local government helped drive down the infection rate, but we are by no means clear of the infection, with several cases popping up each day. However, ever so slowly, the life is beginning to return to a new normal. Beaches and parks are opening up. Restaurants are reopening their doors. I would like to think that the Aloha Mask contributed in a small way to the success. We wound down our operation, but we're ready to spring back into action if the need arises.
Well, let me ask you about that. If I can interrupt you, Serge, let me ask you about that. By the way, your presentation is extraordinary, absolutely extraordinary. And let me ask you, with talk now, especially stateside, of a recurrence, and as you can see, cases are spiking in states across the United States. Are you prepared now? I take it you've taken kind of a hiatus at this moment in producing the masks, but are you prepared to go back into business right away if you have to start making these? And tell the audience, tell all of our ARCS members how many people you actually have who are out there that on one phone call you can get them both activated again. And are you prepared to do that?
Absolutely. So the main reason we wound down wasn't because of the lack of volunteer effort or the lack of materials. It was simply that I think for the island of Oahu, our needs got met. The local hospitals didn't seem to be overwhelmed, and we had about 1,000 cases so far total. But at any moment, we have at least 250 people who are ready to go as soon as we.
Do you have one of the masks right in front of you there?
I do.
Why don't you tell us? Let us take a look at it.
So here's.
Oh my Lord! Look at that! It's not only a mask, but you've designed these things. Who designed them?
So we started from a flu mask pattern that was available on the internet, and then we modified it to include a filter pocket that you can slip a makeshift filter.
Yeah, hold it up nice and high so everybody can take a look at it. There you go. So you put a filter pocket in there as well. Now, how does this compare to the masks now that CDC is recommending, the 3-ply and whatever? You're quite in standard with those, aren't you?
Very much so. Nothing beats an N95 respirator, and our atmospheric tests has shown that. Below 1 micron, you really have to have nonwoven synthetics, which are in such short supply. However, above 1 micron, we do pretty well. We do comparatively to the N95 respirators and about as well as surgical masks.
Well, you've certainly brought a lot of credit to ARCS, who gave you a wonderful scholarship or a grant, but then, of course, little did they know you were going to become an entrepreneur of face masks, literally supplying the entire Hawaii. How many volunteers altogether working on your staff?
We had 400 volunteers sign up. I don't think we were actually able to cycle through all of our volunteers when we were making 10,000 masks.
But how about the drivers too? You said you've had drivers all over Hawaii.
Yeah, absolutely. So because of the lockdown order, we were trying to not become a hub of the infection ourselves. So we had a select group of drivers who had more substantial personal protective equipment and stricter guidelines who were able to deliver these masks. And we had about 50 of those.
One last question for you, if we can, Serge. How much does it cost to make one of these?
In the end, without the labor costs, because it's all volunteer work, it was about 50 cents a mask.
Wow. And you've produced how many now total?
10,000. I mean, take that with a grain of salt, right? Every piece of materials that we acquired was via donation. So if you never realize how heavy fabric is until you have to carry 1,000 yards of it.
Well, congratulations. I must tell you, that is really fantastic. And now for a moment, we're going to switch over to Sanjay. There you are, Sanjay. We see you. Now, for those in our panel or in the audience who have taken that famous and widely given COVID-19 test, it's not the most pleasant experience. A number of our photojournalists and what's been going on lately on our NBC station have had to take it, and I can tell you it's not the most wonderful thing. But now, thanks to Sanjay and his colleagues, the world of COVID-19 testing could be undergoing one of the most extraordinary, inexpensive, and rapid changes ever. Sanjay, what are we talking about?
I think we're talking about something called this dry swab method that I've been working on with my colleagues here. So just for introduction, I'm Sanjay Srivatsan. I'm an MD PhD student at the University of Washington. And here at the University of Washington, we had this thing called the Seattle Flu Study, which was funded by Bill Gates. And back in February and in all of 2019, we were screening for different kinds of respiratory viruses present in the community here in Seattle. And when the COVID-19 epidemic hit, this group basically had the idea to start testing for the coronavirus. And our group actually found the first US case of community spread of coronavirus. So at that point, it became clear that testing, especially of asymptomatic community members, was going to be a big part of getting back to work. And what I wanted to do was take that and break it down into its component parts. Like Bill said, testing means a lot of things to different people. For people that have taken it, it means pushing a long swab all the way to the back of the throat, and then bringing that out and then sending that off to some laboratory. So what we did was we took that and we asked, what are ways that we can reduce the number of parts that are actually needed to do this test and actually change how painful and how invasive the testing is. And the long-term idea for what we want to go for is having everyone do tests at home and then collecting these tests and then putting them into a central place where we can use robotics and machinery to do the actual molecular testing. So I actually brought a kind of the swab that we're talking about. So what we're imagining is something that looks like this. I don't know if you can see that.
Yeah, it just looks like a very small little straw. That's all there is to it. Hold it up again,
right.
Sanjay, so people can really take a close look at it. Yeah, spin it around a little bit. Let people see it. So that's all it is, huh?
It's just a bit of a mascara brush. This isn't the final design. So this is an injected molded version of something we're thinking about. And the idea would be to just kind of stick it in your nose like this, swirl it around. And then
when you think of what's out there,
you kind of distribute small tubes like this right here. And then you can break it off, and then that's it. And then you can kind of send this back to a laboratory where we can kind of rack it and do the testing on it.
Well, let me ask you this, if I can, Sanjay. How would you rank that as far as being effective? Now, there's been a lot of talk about just the effectiveness of the COVID-19 test at all, the standard one, which, as you have said, and I've experienced with other folks, it's not the most pleasant experience in the world, but its reliability has also been questioned. Now, what is the reliability factor on yours? Because you can see it's painless. You just put it in your nose, take it out, and that's it.
We need to do more testing, but I think there's a question of if we're going to be administering millions of tests per day, right? If we're going to scale to that level, how sensitive does a test actually have to be? So if we're missing things at the low end, that could be okay if we're actually catching all the people that are actually spreading the disease and administering these tests. Does that make sense?
What kind of response have you gotten across the world to this thing, which of course is very new and very innovative, and more importantly, very simple? Yeah,
people are adopting it everywhere, actually. So I mean, one thing to note is that this kind of method was already, we were working with a group in Wales to actually develop this. And the idea is really simple. One other part of the innovation of this actual method is that once you get back into the lab, what people normally need to do is extract something called RNA from within the virus to actually do the molecular testing. One other part of my research has actually been to take that RNA extraction step completely out of the picture. So by using a single buffer, which is widely available, we can break that virus open and directly do the molecular PCR off of that. And people are starting to adopt this for saliva. They're starting to adopt it for gargling. It's actually pretty amazing. We actually have a paper out online about how to do this and our preliminary results.
Well, you know, when you started out as an ARCS scholar and you were doing all of your research and everything, what were you working on, and then what motivated you to completely change gears and actually develop something which is quite unique?
Yeah, so I work on something called single cell sequencing. What we do is we take single cells and we sequence the RNA from them and use that information to understand what the cell is doing. So at a very high level, it's a very specific version of the same PCR testing that we do for coronavirus. So when the pandemic actually hit in March, all of our labs shut down, and I realized the only way to be in the game was to actually be part of this effort to help scale testing and put those skills that I'd been training for something more complicated towards something that's more broadly applicable.
Can you give us an idea of when we might see that swab test, the dry swab test, actually out and being used here in the United States?
So our study just started back up again, and we're talking about, you know, as early as this summer, starting to distribute these to different companies or within the University of Washington. Just because the testing right now is very decentralized, every single institution and locality is taking their own approach to testing. So hopefully something like this takes form and we can administer the number of tests we need to get things going again.
Have you had any contact with Bill Gates? You indicated his name to begin with. Have you gotten him interested in this, hopefully?
Yeah, I mean, he's seen the data. He's pretty excited about it. I haven't talked to him directly. My bosses have. But I think he's got the right idea in terms of where the testing needs to be.
So there's a very good chance that he might end up backing you and helping you to get this thing distributed across the United States.
Oh yeah, to be clear, he is backing us, and he is, you know, we're a bit of the laboratory of seeing what can happen, at least here in Seattle. And if that really takes hold, hopefully we can scale it.
Well, we wish you all the luck in the world. What you're doing is absolutely fantastic. And you know, where your colleague where Sanjay invented the mask, and here you've gone inventing this, not inventing the mask, but making it into something that's become so worthwhile in Hawaii. And now you've come up with something here for testing, which is extraordinary. And you know, talking about testing, we've heard a great deal about antibody testing. Millions are doing it and hoping that they have a built-in immunity to COVID-19. Now, Melanie, it's your turn up. We'd like to ask you that you and your team have turned attentions from studies of the immune system and protein interactions, which is what the ARCS members who sponsored you and who led to getting your grant were very much interested in at the time. You've completely changed gears, and you've gone to the critical task of antibody testing using patient saliva samples. Quite unique. Can you tell us what this is about, what you're doing, and if it's working?
Yeah, absolutely. Thank you, Bill. So yeah, a couple months ago when the pandemic really hit in Los Angeles, which is where I am currently getting my PhD, my research lab shut down. And so, you know, my background is really in looking into protein interactions and cell signaling of immune cells. And so I have a pretty good understanding of, you know, proteins and antibodies and immune cells and immune interactions. And so I took it upon myself to find organizations within this community who were, you know, trying to contribute to the efforts of helping solve the COVID problem. And one company in the greater Los Angeles area became really, you know, responsible for handling a lot of the molecular testing that Sanjay was just describing. So, you know, collecting patient samples and running a PCR test to look for any of the viral material in those samples to detect, you know, whether someone was positive for the virus or not. And so this company has really unique access to a large database of people in the Los Angeles area who have, you know, either tested positive or negative for the virus. And this company had been responsible for setting up a number of drive-through centers where people could actually just go in person and take the test and drop it off, and then 24 hours later they would get their results, which obviously is extremely helpful for, you know, detection and letting people know what's going on. And so that group wanted to expand their testing from this molecular testing that we've heard a lot about to antibody testing. And the reason that antibody testing is different is that, you know, people don't actually develop antibodies right away for a virus that they've become infected with. It's more of a long-lasting response to a viral infection or some kind of other infection. And so with the antibody test, we have a way to detect if someone had been exposed to the virus. Maybe they never, you know, went and got a molecular test to determine if they were positive at the time, but the antibodies would tell them had they been exposed, had they developed an immune reaction to this virus. And the reason that that would be important is that some people think that having an immunity against the virus would prevent any further infection. And so as part of this company, which is Curative Inc., we are working to find out if that's really true, because we have access to all these patient samples that really tell us that we can monitor and over time and look at what kind of antibody response remains after, you know, 2 months after infection, 3 months after infection, a year after infection. Those are the kinds of questions that we're trying to answer.
So right now you still have the questions. And so that leads to my question, just how reliable are these tests? I mean, somebody gets the test, comes back negative or positive, just how reliable are they, Melanie?
Yeah, it's a great question. It's a really important question. It's one that we've also been trying to answer as part of our efforts. So we've been taking actual patient samples and testing a number of the available antibody tests that are on the market and putting out some numbers to show that a lot of these tests are really not as effective as they claim to be, or they're not as effective once they get scaled past, you know, just a small clinical study and actually get used in a larger population of patients. So yeah, so unfortunately, as far as we can tell, antibody testing is still, you know, not optimal, at least in the tests that have been released so far for COVID. And it's unclear if that's because patients aren't, you know, like developing antibodies against COVID as consistently as maybe we would expect, or if that's the tests that are available are just not optimal yet, because there really hasn't been a lot of time that has been dedicated to developing these tests. I mean, COVID, you know, has only been around for less than, you know, 7 months or so, as far as we know. And scientists tend to take a long time to really optimize tests like these, in my opinion.
Are we in for any surprises if we go for the antibody test and we think, hey, we're going in there for COVID-19, but of all of our guests who are all over the United States and our other scholars who decide, hey, I think I'll get that done, are we going to be surprised that we could find something out that we don't expect?
You know, I think that antibody testing is still really important, because if you do test positive, most of the time that result will be accurate, an accurate positive test. What that means for you is that, you know, you have had exposure. Whether it means that you're going to be immune to the virus if you do come into contact with it at a later time, it remains to be seen. But in terms of, you know, will you be surprised? I think, you know, people tend to expect that that knowing if they have antibodies will kind of make them feel safe or something like that. And I think that maybe what I would like for people to know, right, is that COVID is a virus, SARS-CoV-2 is a virus that we really still don't know everything about. And so we need to, you know, make sure we still take all precautions against the virus and make sure that we're keeping ourselves as safe as possible, regardless of what kind of results you get from any of these tests.
You mentioned one thing that I think is very important. You mentioned exposure. And I think that if we have the antibody test, and this is just as a caution to everybody out there, if you take the antibody test now, it doesn't necessarily say that if 3 months from now or a month from now in the summertime, as this thing is now spiking again across the United States in many places, that you could still get COVID-19, even though if you do show the antibodies, is that a guarantee, or and you show you've got the antibodies, or could you still in fact be exposed, as you said, going down the road maybe a month or so?
Right. I mean, so that's one of the main questions that we're trying to answer here as part of our effort is, when you develop an antibody, does that really make you resistant to infection, to a secondary infection? And so that kind of, that study has not yet been complete on a scale that I think would give enough results to really be able to make a decision about that.
But you've got nothing to lose by taking it, correct?
Yeah, definitely not. I mean, it's still valuable to know if you have antibodies. I mean, especially in the context of contact tracing, to be able to understand, you know, like, I was definitely exposed at one point to this virus, and find out, you know, how many people you were in contact with at that time, and, you know, what kind of response you had. And a lot of people just in general are curious about whether they've been infected. And I think it's it's definitely a way to find out if you truly were.
Well, there's really no way we can thank Alexandra or Sasha and Sergey and Sanjay and Melanie for all of your time. You have been wonderful. I think you've given your fellow scholars across the United States who are currently watching this, and your members of ARCS who support you 100% and back up everything you're doing. I think all of them are absolutely blown away, as I certainly am, as to the time and effort you are putting into the most innovative and extraordinary work on behalf of all of us. The COVID-19 pandemic that's currently spreading and now coming back to a certain degree. You're all to be congratulated. You're a marvelous, very inspiring group of scholars. And now I would very much like to turn it back to Nancy Chambers, who is your Vice President nationally of ARCS, who really has set this up. And I think she's going to take it here with some questions that a lot of folks out there have. Folks, thank you so much. You've been very inspiring to me. I've loved every minute of it.
Thank you, Bill. We do have a few questions for the panelists. And let's see, the first question is for Sasha, and it has to do with the production cost of the ventilator. What can you tell us about that, and how much will it cost to produce your ventilator?
So Nancy, that's going to partially depend on who we can find as our production partners, and the final form of the ventilator when we do ruggedize it for these extreme point of care environments. I can tell you right now, our cost is less than $150. I think on our website, put a breakdown of all of our materials currently, and we try to keep it as low as possible using a differential pressure sensor as a flow sensor as well as a pressure sensor. We try to using a laser cut acrylic for our box, but I think if we can find the right manufacturer, we can get that down to you know, really definitely under $30 for this type of unit. So don't have the specs on exactly the cost of goods yet, because we're still working on making it something that can withstand things like power surges, the low infrastructure in extreme point of care environments. But at the moment, it's already much lower than a lot of the ventilators out there today, significantly lower.
So, Sasha, a follow-on question from the audience is whether or not you have tested on dummies and artificial lung.
We have done some testing on artificial lung, but not on, and we have a setup here. Maybe I can kind of show you our lab setup here, where we have our connected, this is just in our laboratory, little dummy setup here. But really this needs to be tested much more rigorously, because ventilation in particular can be very dangerous. So it requires thorough testing, thorough characterization of all of the parameters of the volume, the ratio of inhalation and exhalation, breaths per minute, and the pressure. So we're working on characterizing all of that as much as possible, and hopefully we can get this out for further testing over the coming year or so.
Thank you, Sasha. So I have a question from the audience for all of the panelists, and it has to do with where your education and this early scientific experience will take you in your careers and what your aspirations are. And Bill, maybe can you please call on a couple of the panelists to give us some insight into their goals?
Yes, Sanjay, let's start with you. If you could also, just before you answer that, could just ask you, because the cost factor came up, I thought that was quite interesting with Sasha. How much does it cost for you to produce this small little dry tab as opposed to what it now costs to do a COVID test?
Yeah, so that small dry guy is made through a process called injection molding, and companies do this all the time. So I think the cost of that is like 5 cents, and then the actual molecular
as opposed to the COVID test.
Yeah, the molecular parts are dollars. So I think in total, in terms of without any labor, it's probably like $10 per test.
Now let's ask you, if we can, that question about what lies ahead for you. What's your dream job? Where are you going in your career after COVID hopefully quiets down and hopefully goes away?
I'd love to have a research lab at a kind of R1 university. I mean, the virus has totally shifted people's timelines for different things, and kind of changed short and long-term prospects. So we'll kind of just have to see as we go.
Serge, if we can ask you to put down the mask for a moment and tell us, what are your long-range goals? Where are you going to go from here?
Of course, Bill. I actually just finished and defended my PhD on March 12th. So about 2
Congratulations.
weeks. Thank you. 2 weeks before
just
the university.
just in time to make the masks.
Yeah, absolutely. So I went from having long nights of writing my dissertation to having long nights of working in front of a laser cutter. I work in private industry doing scientific research, and I'll probably continue doing that for the foreseeable future. Probably not planning on making Hawaii my home, and I'll be returning back to the mainland probably within a year.
Well, make sure you take that shirt with you. That's a great reminder of where you're living now. And Melanie, can we ask you, what lies ahead for you? What are you thinking about after you are able to get through all this diagnostic testing and antibody testing?
Yeah, of course. I mean, diagnostic testing is really interesting to me, but I'm still a second-year PhD student, and so, my research, while it is immune-related, is not quite in the same diagnostic field. So I will likely see through, a couple more months of COVID response, and then go back to my PhD. And like Sanjay, I think one day I would like to be, the director of a research lab, whether that's, at a research institute or university, is still to be seen.
Maybe we have just 1 minute to ask somebody real quick. I would like each of you just to tell us, if you have one last thing you want to leave for this marvelous audience of fellow scholars and for members of ARCS across the United States. Serge, we'll start with you and just work our way down. One last thing you'd like to say real quick in 15 seconds.
Absolutely. We documented all of our effort on alohamask.org, and if anybody wants to follow in our footsteps, if you want to make 1,000 masks a day, it's all there.
Alexandra, what about you? One last thing to tell folks.
I just wanted to thank ARCS for all of the opportunity that they've given us and inspired all of the scholars to be able to take these initiatives and make these types of devices. I think this crisis has put to the forefront that more and more we do need funding in and investment in science. And I think it's also putting to the forefront that, you know, this kind of decentralized, personalized, portable diagnostic, portable medicine, personalized medicine is the future. And I'm very excited about hearing everyone's work in this area.
Melanie, what about you?
Yeah, I completely agree. I mean, thank you, ARC, so much for everything that you've given me and done for us in, you know, the past couple of years. And what I would like to say, I guess, is that, you know, it's just important to be patient with scientists in this time. I know it's tough to, you know, not be able to go back to regular life as things were before the pandemic, but scientists are working, you know, as hard as they can and as quickly as they can to get, you know, vaccines on the market, get diagnostic tools that work onto the market. It's just, we're all working a lot faster than we're really used to. So there are still hiccups that are part of the process, but, you know, we'll get there sometime soon.
Sanjay, what about you?
Yeah, I want to echo what Melanie and Sasha said. I want to thank ARCS for funding us. I just hope everyone stays safe out there. You know, like, you know, I think what Serge is, I mean, just, you got to wear a mask, you know, be careful when you're indoors with other people. I'm very worried about a second wave, and I really want everyone out there to be safe.
Well, thank you all very, very much. Nancy, back to you.
Okay, thank you. And thank you, Bill, and panel members. We have a few questions that we weren't able to get to, and the panelists will answer those over the weekend. They'll be published in our newsletter that will come out on Monday and Tuesday of next week. And so keep an eye out for that. They'll have a little more to say about their work in that newsletter. It has been inspiring to hear about all of this work that these great scholars have done. And we want to mention, though, that there are many more scholars and alumni who are making significant contributions daily in this fight against COVID. So before we end, we want to recognize a few of those scholars who have done such an amazing job. And let me just show you their pictures here. Having a little bit of a technology delay. Can you see my screen, Bill?
Yeah, we sure can. And we see what has now become the COVID-19 famous virus picture. So
Okay.
Hopefully the scholars' pictures will be coming up. And I see that Serge over there has got his mask on because he's not sure what's coming up. He wants to make sure he's protected over there. And luckily, Alexandra's not having to use the ventilator. And Melanie's got a smile on her face. And I know that Sanjay's down there somewhere. I just can't see him. I know he's down there. There he is. And there's Sherry also. Yeah, no, the pictures are not coming up, unfortunately, at the
Okay.
moment.
I do have several, though, whose names I want to mention. I want to tell you about Kelsey Lieberger, who is actually she's a first-year ARC scholar. She's done some extraordinary work in COVID data analytics. She and a colleague were the first to apply the Imperial College London model that was validated in Europe to estimate the severity of the US epidemic. Their early work revealed the true number of infections in the United States. And it showed that it was 21 times more than was being reported by others. Since then, antibody testing has broadly validated her findings, and she has a paper that has been posted on medRxiv. When I asked Kelsey what enabled her to be the first in the world to predict what was happening in the United States, she cited speed and how important that was. And without hesitation, she said that her ARCS award had made a big difference in her being able to complete this analysis in an unusually short amount of time. I'm going to see if I can bring up my pictures now. And so bear with me here. Let's try this one more time and see if I can show you some of these scholars that we want to recognize. Another one is Erin Murray, who is a, here's Kelsey, can you see her?
Yep.
Okay,
Yes, she is.
great. And Erin Murray.
Yeah, there she is.
Okay, wonderful. She's doing significant work serving as the epidemiology team lead in the California Department of Public Health COVID-19 response. Her department has tracked more than 133,000 COVID-19 cases to date. She manages a group of epidemiologists and data managers who conduct surveillance and report their findings to the Director of California Public Health and the Governor of the state of California daily to inform policy in California. And our next, this is an alum that we want to recognize, Dr. Erin Newman. She's actively involved in the COVID-19 response, both clinically, caring for patients in the coronavirus ICU, and in research, which includes the clinical coordination and design of the Seattle flu study. She posts about her work very frequently. She does a great job of curating very significant work from the enormous amount of information that is published daily on COVID. And you can follow her on Twitter and many of these scholars as well. And the next scholar is Gabrielle Pastinko. She is doing COVID-19 surveillance and research at the California National Primate Research Center, working to develop treatments and a potential vaccine. We have a team at the University of Georgia, a scholar and a scholar alum. Kate and Cecilia are working together as a team as members of a larger COVID-19 portal team. Their role in this massive undertaking has been to analyze comparative data that contrast characteristics of coronavirus and other infectious epidemics, and to develop data visualizations that make the data easy to understand. And you can access their portal with covid19.uga.edu as the address. Finally, the last that we're going to recognize today in this event is Fred Streitz. He's currently serving as the Department of Energy liaison to the FEMA Data and Analytics Task Force. He's helping coordinate all of FEMA's computing resources to aid in the COVID-19 response. Dr. Streitz has had a remarkable career as Chief Computational Scientist for Lawrence Livermore National Lab, and he will be featured in the August issue of our national newsletter, so you can read more about him then. In fact, you can read more about all of these scholars and our panelists and many other scholars that we did not have time to recognize today who are doing important and interesting things that relate to COVID on our website under ARCS Scholar Achievements, our ARCS Scholar COVID Achievements. Sherry, I will turn it back to you.
Thank you, Nancy. And I want to thank all the ARCS scholars and alumni for taking the time to present today on our first scholar panel. Thank you, Bill, for moderating the panel, and thank each of you for joining us today. I hope you found the time you spent with us informative and that you were inspired by these young scholars and alumni. Remember to tweet and post about today's event, and please follow us on ARCS social media platforms.